Hello, everyone. I'm Lisa Ling, and welcome to the second episode of The Road to a Vaccine, a weekly live educational series exploring the COVID-19 crisis and the global community's efforts to develop a vaccine against the disease. Last week, we heard from scientists on the front lines. We talked about what a vaccine actually does and that many different types are being developed by different researchers. But perhaps the most striking thing we discussed was the opinion expressed by public officials on the show that there really is no path to ending a pandemic other than a vaccine. Now, as of this morning, Johns Hopkins shows there are over 2.5 million confirmed cases worldwide and over 170,000 deaths. That's over 50,000 deaths since this time last week. Things are changing every day, and there's still so much we don't know. And that's the reason for this series, to help us get some answers and to give you the most up-to-date information from experts on the front lines. Today, we have three of them with whom we will be talking live. We've also had an outpouring of questions from viewers last week, and I want to get to as many as possible. So put them in the comment section, and I'll be taking your questions throughout the show. Today, we have two researchers who've been working together across COVID vaccine. First up, we have Dr. Dan Barouch, professor of immunology at Harvard Medical School. Dr. Barouch, thank you so much for joining us. I want to first take you back to January of this year. China publishes the sequence for the virus. What does that mean for you as an immunologist, and what did you do with that information?
Well, thank you, Lisa. It's a pleasure to be here. On January 10th, the sequence of the novel coronavirus was released by Chinese researchers, and we, like many others in the world, started analyzing the sequence that same day. So we started analyzing the sequence of that virus in the evening of January 10th. We worked with our team through the weekend, and by Monday, January 13th, we had isolated or we had designed potential candidate antigens to be used in the vaccine. So the first
That's
few
astounding.
days of the vaccine effort were actually done electronically, just analyzing the sequence that had been published. So that was the starting point.
That's absolutely astounding. And in what ways did you collaborate with other scientists on this?
Vaccine development is intrinsically collaborative because there's not a single group that can do everything, from basic discovery to large-scale manufacturing, to animal studies, to clinical trials, to eventual delivery of the vaccine. So vaccine development intrinsically is a collaborative endeavor and requires people from many different disciplines. So we have been fortunate to collaborate with many groups, as well as with J&J and other groups of individuals in our efforts to develop this vaccine.
So it was obviously determined that this was a coronavirus. What exactly is a coronavirus, and are there unique characteristics about it? And how is it different, this one, from other coronaviruses?
So coronaviruses is a class of viruses. It's not a single virus. And there's examples of coronaviruses in animals, and there's examples of coronaviruses in humans. The human coronaviruses can be divided into those that cause nothing more than the common cold and those that cause more serious disease. There's three examples of coronaviruses that infect humans that cause more serious disease. The original SARS virus in 2003, the MERS virus around 2012, and now COVID-19, also called SARS-CoV-2. One thing that's different about COVID-19 compared with the original SARS virus is that it appears to be much more contagious, much more transmissible. In addition, there's asymptomatic spread from individual to individual, which in SARS was thought to be very minimal. In fact, most people with SARS had symptoms. So overall, the clinical disease for COVID-19 is less severe than for SARS on an individual basis. But it's that very fact, the fact that many people are asymptomatic and still able to spread the virus, that leads it to be much more difficult to control. And that's why this has emerged as a global pandemic.
As you started learning more about COVID-19, did you think this virus would become one that would destabilize countries the world over?
In early January, when we decided to start working on this virus, then we were concerned that it might have pandemic potential because of its explosive spread in China And because of the possibility, later clearly proven, at that point it was a possibility that it could be spread by asymptomatic individuals. Because of that, we were concerned it had pandemic potential, but we did not know at that point in time it was going to evolve into such a dramatic worldwide pandemic and truly destabilize economies and countries.
So let's talk about the vaccine candidate that you helped identify. What is significant about this vaccine candidate that you've been working on?
So for this vaccine candidate, we're using a common cold virus that is made to not replicate in humans, so it can't cause disease. We're using a common cold virus as a carrier, a shuttle, also called a vector, to bring in a piece of the COVID-19 virus, not the whole virus, but just a very small piece encoding the key spike protein. We're using the common cold virus to bring the coronavirus spike protein into cells so that the human body can make immune responses to it. We and others have shown that this is a very effective approach for raising high titers of antibody responses as well as T cell responses. And in humans, those can last a very long time. So we thought that this platform technology would be ideally suited for this type of a vaccine.
Now, what happens after you find a lead candidate and it has an immune response?
So once a single lead candidate is identified, then it can go into large-scale manufacturing. This is where another important feature of this platform is. J&J has developed manufacturing platforms that are able to scale this up to a very large extent, so that millions and hopefully even 1 billion doses of vaccine can be manufactured with this platform. So after a lead candidate is identified, then clinical manufacturing of the vaccine has to proceed before clinical trials can start.
Dr. Barouch, I have a question here from Stuart from LinkedIn. With all the news about antibody testing, is this something you can really determine if we get the virus and how bad?
So a good antibody test is critical for charting the spread of the pandemic worldwide and to evaluate how far it's spread already. We actually don't even know today the number of individuals that have been infected because typically only symptomatic people are being tested nowadays. So it's possible that the number of people infected is actually vastly higher than what the current numbers indicate. However, we have to be careful with tests because we have to make sure the tests are accurate. And right now, there's emerging multiple different tests, and we don't yet know precisely which are the optimal tests to use. So there will need to be a lot of work in the diagnostic space as well.
We have another question here from Christine from LinkedIn. Has an effort like this with all scientists collaborating together ever happened before? And if not, why?
I think the global need and the intensity of COVID-19 research as a whole, as well as COVID-19 vaccine work in particular, is unprecedented. There's more open sharing of information and data and ideas than I've ever seen before, because ultimately we're all in this together. And we all want the same goal. We all want to develop a vaccine that can end this pandemic. So many structures, many collaborative organizations, many phone calls are really promoting a lot of sharing amongst individuals and collaboration, because ultimately collaboration is key to making this vaccine successful.
Dr. Barouch, you have worked on Zika, Ebola, RSV, HIV, and now COVID-19. What are the similarities and differences in the viruses? And does that change in the way that you make the vaccine?
So for COVID-19, the global need and the urgency and the intensity is far greater. There are some differences also worth mentioning. For HIV vaccines, there are some major challenges, such as the degree of virus variability and the lack of any known immune protection correlates, and the fact that no one for HIV has spontaneously cleared the virus based on their own immune responses to the best of our knowledge. For COVID-19, we see some clear differences with HIV. Although there is some virus variability, overall, there is much, much less virus variability for COVID-19 than for HIV. In addition, most people who get COVID-19 recover. And so while we don't yet know for sure that they're resistant to reinfection, we do have substantial belief that the human immune system is able to recognize and contend and clear this virus. So based on those arguments, we have experiments of nature suggesting that a COVID-19 vaccine is likely going to be technically possible. So COVID-19, although it has the intensity and urgency that some previous vaccine efforts didn't have as much, there are also some scientific issues that make us more optimistic about the potential of the development of a COVID-19 vaccine than, for example, for HIV.
Incredible. I have one more question from April from Facebook, who asks, can you tell us about the possibility of using a biologic to treat COVID patients?
So there are many different treatment modalities that are being advanced and studied for COVID-19, both small molecule drugs as well as biologics. So by biologics, I assume you mean monoclonal antibodies. Many groups are developing monoclonal antibodies for COVID-19 that are going to be studied both for prevention and therapeutic purposes. People are already studying plasma from convalescent patients, which is also a biologic. So there is a lot of work in that area, and there will be many results of clinical trials of both small molecule drugs as well as biologics in the coming months.
Dr. Barouch, why does the world need a vaccine?
Well, the explosive spread of the virus together with asymptomatic spread suggests that a vaccine might actually be needed to end this pandemic. And every day that has gone forward from January till today, in my opinion, increases the likelihood that a vaccine is going to be the ultimate way of ending this pandemic.
And the numbers are just continuing to grow so exponentially day by day. I want to thank you so much, Dr. Barouch, for being with us. In case you're just joining us, I'm Lisa Ling, and this is The Road to a Vaccine, a weekly exploration of the COVID-19 crisis and the global community's efforts to develop a vaccine against the disease. I want to remind everyone to ask questions in the comment section, and I will hope to get them answered throughout the course of the show. Our next segment is one that we are calling Vaccines 101, which is a deeper dive into the basics of the vaccine process. Now, last week we talked about what a vaccine does, and now we want to talk about what it takes to get it into clinic. So our guest today is joining us from the Netherlands, where she is the global head of viral vaccine discovery and translational medicine at Johnson & Johnson's Janssen Vaccines. Dr. Hanneke Schuitemaker is a trailblazer who spent her career tracking the HIV vaccine code and now coronavirus. Hello, Dr. Schuitemaker from the Netherlands.
Hi, Lisa.
Hi. On the show last week, we talked about how a vaccine candidate has been identified. So now what? I know scientists are working around the clock, but what exactly are they working on?
Yeah, so the scientists are indeed working around the clock. We want to get more data in animal models to see how our vaccine candidates are performing. But in parallel, we are preparing now for the manufacturing. So that process has started as well. And that brings in a complete new army of people that are doing the test runs for the vaccine manufacturing. The clinical teams are gearing up. They are making the plans on what the clinical trials should look like, where we want to do that. We need the assays to be able to read out the responses in people. We need the interactions with all the regulatory authorities to see if our plans fit their needs and if we agree on how to move forward. So it's really a busy time.
Can you tell me about this army of people that you refer to? How many people are working on this effort, and what kinds of roles do they have? Kind of take us inside to the inside the world a little bit, if you can.
Yeah, sure. So of course we have all these layers, and sometimes people think that that's too much, but everybody is so busy, so everybody is needed. We have people who are doing the pipetting in the lab, who are doing the construction, building assays to be able to see if now animals respond to our vaccine candidates, and later on whether people respond in the right way to our vaccine. We have people who are running the small-scale bioreactors to see if the manufacturing looks good. We have people already running larger-scale bioreactors to see if that goes well. So we are doing a lot of things in parallel that normally go sequentially. And of course, we also have our strategy ready to see what do we need to measure in people to get a level of confidence that the vaccine may work. Because of course, everything is still theoretical, right? We have absolutely no clue if this vaccine will be successful. So we really want to do the right things to become a little bit more certain of what we're doing.
That's the thing. There still is so much that is unknown. And we've been hearing about a number of companies worldwide that are working on vaccines. So can you talk about the landscape and how many different vaccine methods are being tested? Why is it important to be trying so many different approaches?
Yeah, so I really think it's important that we do not put all our eggs in one basket, right? So it's really good to make use of different platforms. And what you now already see is that some of the platforms are more rapidly able to move into clinical testing because that first phase for them is faster. But we will learn so much from these front runners, and maybe we can leverage knowledge that they gain with their studies to maybe accelerate other candidates that are in the making. So I think that the RNA candidates are going fast. They really use genetic material as a vaccine. We also have the more traditional vaccines that are based on the protein of the coronavirus, and they use then an adjuvant to enhance the immune responses to this protein. And of course, we have the vector-based vaccines, and that's one of Janssen as well, where we put our money on to see if that can work. And based on the experience we have with this platform, this vector system in programs for HIV vaccine candidate, for RSV, for Zika, we know how it performs, and also we know that because the outside is always the same, that vector looks the same, we know how the human body will respond to it, and we know that people tolerate our vaccine candidates quite well. So those are good things to know to move faster in clinical development. The regulatory authorities know our platform, and they also have a level of comfort. So it really facilitates the dialogue of going fast. And I assume that others have that benefit as well. So that's why it's good to have multiple candidates to see which one will be successful and how we can move fast as a field, not necessarily as a company.
You have such extensive background in vaccine development. Have you ever worked this fast?
No, and we are getting better and better at it. So when we were dealing with Ebola, we were really challenged to be for a first in human study within 4 or 5 months. We already had a vaccine candidate ready at the time because we were working on that already for quite some years. And we managed, and we were so happy that that went well. Then Zika came, and we managed to be in the clinic between conception of the vaccine candidate to first in human study in 9 months, and now we are trying to beat that by doing it in 8 months. And so it's not cutting corners. It's really getting more familiar with the plug and play platform that we say we have, and with the knowledge of the tolerability of our vaccine platform, that really helps to go fast. But I think this will be a hard to beat timeline if we manage to be in the clinic in August.
I have a question from Bhushan from Facebook, who asks, is developing her immunity the only way till we will have a prophylactic vaccine?
So I think that a prophylactic vaccine will help to build herd immunity if we have to go through this waves of infections to get a sufficient percentage of the population immune so that these waves will stop. That will take years. If we have a good prophylactic vaccine that can protect people, I expect that the herd immunity can build much faster. But we indeed need multiple, I expect multiple good working vaccines to establish that much more rapidly.
Now, throughout history, vaccines have really been the only solution to ending a pandemic, as with smallpox, measles, and Ebola. Why is that?
Yeah, so I think the most critical piece is that people, that with vaccine, you can stop the transmission chain. So now in this current situation, people can transmit virus when they are not yet symptomatic, right? And that makes it very difficult to quarantine people on time to prevent transmission. If you can vaccinate and make sure that people become non-transmitters so that they do not develop viral loads sufficiently to infect somebody else, then you can only get that herd immunity effect, that the virus cannot be passed on. And that is what vaccines do. So treatments can save lives, but vaccines can save populations.
So my husband is a physician and a scientist, and he says that COVID-19 has been mutating fairly slowly. Do you have a perspective on that? And how can scientists account for mutations of the virus when developing it?
Yeah, it's indeed a good thing that the mutations are limited and appear slowly. That gives good hopes that the virus will also not be able to escape immune responses, and that also a vaccine may have a prospect of working for a longer time if we and others are able to demonstrate effectiveness of our vaccines. The reason why it mutates slowly is apparently that gives the virus its fitness, because if it would be non-beneficial, it would be sloppy and would make mistakes. So there's always an evolutionary reason for a virus to why it is what it is. And so I think with the current mutation rate that we see, that we will have a good prospect of getting that herd immunity in place. And of course, we are testing whether our vaccine will cover new variants that may arise, but that's not yet an issue.
Dr. Schuitemaker, I have a question from Rohan from LinkedIn, who asks, is J&J working with other companies working on their own vaccine candidates to speed up development?
Yeah, so we are in several public-private partnerships where we discuss with other manufacturers on how we can learn from each other and how we can leverage that knowledge to speed up. Of course, everybody is sort of in the same darkness because there's a lot of things we don't know about this virus. We don't know if natural immunity will be protective, right? Or antibodies that are present after infection, whether they can protect from reinfection, what the levels of antibodies need to be protected. So we need to learn a lot that applies to all of us. And it's really good to have those discussions and to try to help each other and to define the critical questions so that there's a focus on those to help the field forward.
Patrick from LinkedIn asks a question that I think most of us are really anxious to know. Once the human clinical trials begin and are deemed successful, how long will it take to get approval for widespread use?
Yeah, so that approval is not up to the companies, right? It's really the regulatory authorities that in the end decide whether a vaccine is good enough and can be used in a population. As it seems now that all authorities say that you need to demonstrate effectiveness before a vaccine can be used at larger scale. So that is all dependent on how the pandemic develops, how long it will take to demonstrate vaccine effectiveness, and then of course after that how rapidly vaccine doses will be available, and a decision can be made on the safety, the tolerability, and the effectiveness of a vaccine to be used. So if I had a crystal ball, I would tell you, but we don't know yet.
Well, Dr. Hanneke Schuitemaker, thank you so much for taking the time to be with us. So we have some takeaways so far. One, we have a long way to go still. Two, there are many different vaccines that are being worked on now, and it's really important to identify the one that works. Three, as both of our guests have said, this really is a massive global effort, perhaps unlike anything that we've experienced before. And ultimately, we have to follow the science. Now, remember, we are live right now, and we encourage you to write your questions in the comment section, and we'll be asking them throughout the show. My next guest is Dr. Dan Carucci, Global Medical Director, McCann Health. He is a physician scientist with more than 25 years of experience in large-scale innovation programs, vaccine development, and global public health. Hello, Dr. Carucci.
Hi, Lisa.
Hi, I want to get right into this with you. We've been talking for the last 2 weeks about how a vaccine is the only way to stop COVID-19. And frankly, this has a lot of people concerned, particularly people who are just opposed to vaccines to begin with. I've gotten a lot of messages via social media of people who really are expressing these concerns. So how do you convince a wary public of the importance of a vaccine?
Yeah, well, I think, look, vaccines are in essence a victim of their own success. In the 1950s and 60s and 70s, we had the threat of measles and polio and even smallpox. So vaccines were an essential part of growing up. Moms immunized their kids to protect them from serious threats. I think what we're seeing with respect to resistance to vaccination has come because those threats no longer exist, particularly in high-income countries. The situation has changed dramatically with COVID. I think everyone is waiting for the vaccine, and I think there is going to be now a new appreciation for the importance of vaccination. At the same time, I think we have to be concerned and ensure that we develop effective communications to be sure that when the vaccine is available, that we position it as a valuable proposition for populations.
I know you've done a lot of work in Afghanistan, and you developed something called the immunity charm bracelet in Afghanistan. How did those cultural beliefs play a role in the community's receptiveness to vaccines?
Well, the situation of vaccination in the developing world is much different than in, for example, the US or Europe, where not only are there problems with supply chain and access to vaccination, but vaccines may not be positioned as being valuable in a mom's daily life. And so what we did when I was working in Kabul back in 2016 was had the realization that moms placed a small bracelet on the child's wrist, and this protected them against evil forces. And so we ended up harvesting that cultural belief system and generated something called the immunity charm, so that we use the cultural belief system of a bracelet in terms of protecting against evil spirits as a bracelet to protect against diseases through vaccination. And so it was tremendously successful in terms of the appreciation that moms had for vaccinations, and now we're testing it in India through a public-private partnership.
Do you believe a similar kind of cultural shift has to happen with this vaccine that is going to be imminent?
Well, I think in the developing world, I mean, look what happened in Congo and West Africa when Ebola vaccine was introduced. There are still pockets of resistance and mistrust, but I think we're going to have to think about how we develop communications that taps into people's belief systems, their cultural belief systems, to be sure that we don't see the kinds of resistance to vaccination that we saw with Ebola.
And that really is very much what this series is about, trying to communicate with the public what goes into this process of vaccine development, because at this point it affects everyone worldwide. Now, you've spent much of your career working on vaccine development efforts against malaria. Can you talk about your work in this area and some of the challenges that you faced in creating a vaccine?
Well, making a vaccine against malaria is an incredibly difficult process. First, because malaria is a parasite that's co-evolved with us for probably 100 million years. We also don't develop sterilizing immunity to malaria. You can get malaria frequently. most viral infections, like for example, measles, if you get infected with measles and you recover, you're immune for life. So the prospect of developing a measles vaccine was quite easy compared to developing a malaria vaccine because we knew what happened in nature. Making a malaria vaccine meant that we really had to understand the immune responses to the malaria parasite and try to manipulate them and manipulate the human body's immune system to those specific antigens which we thought were responsible for protection. So the challenges that we're seeing with COVID are likely less substantial than they would be, for example, for malaria.
And are you seeing similarities or differences, playing out to how the development process is going with this coronavirus?
Well, I think as Dan mentioned, we think that natural infection does cause a degree of sterilizing immunity. There's much more that needs to be done to understand the human immune response to coronavirus. That portends well for the development of a vaccine. If the body is able to create an immune response which is protective, then being able to replicate that through technology is a good thing. Biology is hard, technology is relatively easy. So I think that suggests that we have a good chance of success for the development of a COVID vaccine.
I have a question from Julia from LinkedIn who's asking, regarding the progression of phases, have you progressed past certain phases toward the development of a good vaccine, and how many phases are there? It's a great question.
It is a great question. Probably best asked to your previous two guests who are actually involved in COVID-19 vaccination, but I can tell you in general, there are 3 phases. The first phase is really the safety testing, using a limited number of volunteers to test for the safety of the vaccine. And safety is incredibly important because you're giving a vaccine to people who do not have the disease. And so you have to be sure that, and particularly if you're injecting and vaccinating large numbers of people, that the vaccine is safe. Phase 2 generally is focused on efficacy. So then we increase the number of patients. We know the vaccine's safe in a small number. We increase the number of patients in phase 2. We look oftentimes for efficacy at this stage while also looking at safety. Phase 3 is much larger numbers of people, maybe in the thousands or 10,000s. I'm really trying to prove the effectiveness of the vaccine in large-scale populations.
Dr. Carucci, we've been talking about this idea of immunity for people who have already had COVID. South Korea has had an incredible response to the virus, but it's being reported that some people thought to have some immunity are getting reinfected. What does that say about the nature of this disease?
Well, I'm not sure that we are convinced that people have been reinfected. It could be that the detection of the virus in those individuals is because the test being used is to detect the RNA. So we don't know that the virus is viable. It may just be that we're able to detect the genetic remnants of the virus in those individuals. It would be unlikely, I think, what we know about the human immune response to viruses, particularly right after someone has recovered from an infection, that they would be able to be reinfected. So we're going to need to do more studies to look into that.
I have a question. A viewer on jnj.com asks, how does modern computing power impact vaccine development?
Dan mentioned how quickly he and his team were able to take the genetic sequence and develop vaccine candidates from the genetic sequence of that virus. He likely was able to compare the genetic sequence of this novel coronavirus with other coronaviruses. So being able to do comparisons at the genetic scale will enable us to develop candidates which are uniquely positioned and which we think will be effective in vaccine development.
And that is what is allowing this development process to happen so much faster than development of years past.
Mm. Yes, absolutely.
So last week, Dr. Carucci, Dr. Paul Stoffels said that we will remember this time as the world before a COVID vaccine and the world after. Now,
Mm.
life as we knew it, even a few months ago, is really no longer relevant. Right now, we can't have an abundance of physical contact with people. So much of the world is now obligated to wear masks. We have to gather in small groups as opposed to very
Mm.
large gatherings. How, or do you have any suggestions on how we can better adapt to this cultural, this really colossal cultural shift?
Mm. Well, I'm an optimist, and I was reflecting recently about the great generation who survived and who lived through World War II. There were a lot of restrictions on society back then, and what came from that, I think, was an appreciation for things that when they weren't available. Commodities in the grocery store, for example, the importance of loved ones. And I really wonder if the focus should be on our appreciation for the things that we've taken for granted. I know in my own personal life, I'm spending more time thinking about conservation, about the importance of my friends and family, staying better in touch with them. I think maybe we have now, there's been a greater sense of community, something that I think we've lost in the past couple of decades. So rather than focusing on the things that we're not able to do, I'm tending to focus on the things that I'm able to do and have a better appreciation for the things that I've taken for granted previously. And I think it's important for us to try to find ways of reinforcing that within communities.
That was a beautiful final thought. Dr. Carucci, thank you so much for taking the time to be with us today. Now,
My
that is
pleasure.
it for the second episode of The Road to a Vaccine. Please keep submitting your questions at jnj.com/road to a vaccine, and we'll be answering more next Tuesday on another live episode. In closing, I'd like to once again thank our guests for being with us today and to all of you for watching. And we all have so much appreciation for those frontline healthcare workers who are just working tirelessly to help people survive this crisis. So I thought it would be nice to hear directly from the workers themselves about just how much the support means to them. We'll see you next Tuesday.
Emergency room nurse in Los Angeles. After a challenging shift, getting home after work and hearing all the cheering for healthcare workers has really lifted my spirit during this difficult time.
Just the outpouring of love and support and gratitude from everyone in the community just means a lot right now.
When they called to check on their loved one and still took the time to ask about my health and safety.
What really got me through my day and every day during this pandemic is the spirit of teamwork and camaraderie.
Hearing the Rocky theme song my hospital plays when a patient comes off the ventilator.
Watching the healthcare workers around me be so adaptable to change and willing to move into areas where the help is most needed.
What lifts my spirits is just seeing the general appreciation for each other and humanity coming together. It's been really amazing and just uplifting and encouraging for everybody.
We will get through this together, and tomorrow will be another day.